You're still muted.
Is the slides are visible? Okay.
Yes.
Okay, great. Thanks very much, Wolfgang, and thanks to the organizers for inviting me to be a part of this series. My name is James. I'm James E. Pustejovsky. Wolfgang, the computer scientist, is James D. So we're in alphabetical order. He's my uncle, actually. So I like Wolfgang said, I'm at the University of Wisconsin-Madison in the School of Education there. And today I'd like to give you a little bit of an intro to some of my work on synthesis of dependent effect sizes using a combination of the R packages metafor and clubSandwich. And what I'm talking about today is mostly drawn from joint work with my colleague and graduate school classmate Elizabeth Tipton. And we've got a preprint available on all of this if you want to get into the nitty-gritty details. So if you have been involved in any sort of research synthesis, you'll probably find this next bit familiar. One very common challenge in research synthesis projects, especially larger synthesis projects in the social sciences, is that it's quite likely that you'll encounter dependent effect sizes, meaning effect size estimates whose sampling variances are correlated with each other. This can occur for a number of different reasons. One common one is that you may have effect size estimates for multiple outcomes measured on a common set of participants. So instead of, say, an experimental study measuring only a single primary outcome, it measures 2 or 3 outcomes, all of which are within the scope of your review. And so you'd like to be able to extract, be able to work with and analyze all 3 of those effect sizes, for example. A similar situation occurs if you've got outcomes measured at multiple follow-up times, say not just the effect size for the immediate post-intervention time point, but maybe also like a 3-month and a 6-month follow-up. Another situation that occurs commonly in intervention studies is you may have a study that compares multiple treatment conditions to a common control condition. So you'll have an effect size estimate comparing treatment T to control, but then also treatment U to control, for instance. And these 2 effect size estimates will be correlated. There'll be correlation in their sampling errors because both of them use the data from the control group. Beyond the context of experimental or intervention studies, dependence issues also occur if you're working with correlation coefficients or other sort of measures of association. You may have a situation where a particular study reports not just a correlation between A and B, but a correlation, also a correlation between A and C and A and D. All of those correlations might be within the scope of your review. And those 3 correlation coefficient estimates will be statistically dependent because they're based on the same sample of respondents. So it's easy enough to extract these multiple effect size estimates from a set of source studies. The problem and the challenge of dependent effect sizes enters in because it's often difficult to get the information that you need to precisely estimate the degree of dependence between effect sizes, to know just exactly how correlated, say, the estimate from say DO1 is with DP1, or to know the degree of correlation between RAB and RAC. The formulas for this sort of thing are available. You can get them in the Handbook of Research Synthesis Methods or many other places. Wolfgang has some functions in metafor to calculate the covariance matrices for some of these cases. But the problem is there's often not enough information reported in the primary studies to be able to use the formulas. For example, in the first scenario where you've got multiple outcomes, you need to know, to be able to estimate the correlation between effect sizes, you need to know what the correlation is between outcome O and outcome P, between outcome O and outcome Q, and so on. And oftentimes that information is just not reported in primary studies. Let me give you just an illustration of how this plays out in a real meta-analysis. Emily Tanner-Smith and Mark Lipsey are both very accomplished research synthesists. They've got a study from a couple of years ago looking at the effects of brief alcohol interventions for adolescents and young adults. It was a very large meta-analysis with 185 studies and a total of nearly 1,500 effect sizes. Each of those effect sizes was a standardized mean difference comparing alcohol consumption outcomes for between intervention participants and comparison participants. So you can see there's definitely a lot of effect size multiplicity here. 1,500 effect sizes in 200 studies. And the individual studies had between 1 and 108 effect size estimates. The median was around 6. The interquartile range was 3 to 12. So there were a few studies that just had an enormous number of effects in them. And that multiplicity arises because some studies measured multiple outcomes, some measured multiple follow-up times, some compared multiple intervention groups to a common control group. Some actually compared an intervention group to multiple control groups, like an active control condition and a passive control condition. So we've got all sorts of dependence. And as is pretty typical, the information that you'd need to calculate the degree of correlation between these effect size estimates was just not available in the primary literature base. So one increasingly popular statistical technique for handling this sort of situation is called robust variance estimation, or RVE. RVE uses a statistical technique, sometimes called the sandwich estimator, to quantify the uncertainty in average effect size estimates or in meta-regression coefficient estimates. And so those variance estimators are used to get standard errors. They're used to conduct hypothesis tests. They're used to construct confidence intervals. The big advantage of RVE is that the sandwich estimator is robust to the modeling assumptions about the structure of dependence in your data. In other words, so even if you get those some aspect of the model incorrect, like you don't know, you're not able to estimate the real degree of correlation between effect size estimates. You can use these sandwich estimation methods, or RVE, to get standard errors that will still work, even if your modeling assumptions are misspecified. It's kind of like you're buying extra insurance on your standard errors and hypothesis tests in case some part of your model is flawed in some way. One of the components of RVE is that it uses what's called a working model to approximate the structure of dependence in your data. It's called a working model because it doesn't have to be exactly right. I mean, that's the whole point of using RVE in the first place, is you want to, we're using methods where we don't want to have to be exactly right. But working models are useful to the extent that they're in the right ballpark of the real dependence structure. So using a working model that gets closer to the real data generating process will give you average effect size estimates, meta-regression coefficients that are more precise than if you use a working model that's not as close to the truth. So it pays to be in the ballpark. But even if you're going to use RVE methods to get sort of insurance and be robust to model the specification. And those of you who work maybe, so if you work in biostats, this technique may sort of ring some bells. In biostats, it goes under the name of generalized estimating equations, harkening back to the sort of seminal paper by Liang and Zager in the 80s. If you work in econometrics, you might know this as just as clustered standard errors. And then if you're a statistician, you know these as sandwich estimators. Actually, if you're a survey sampling fan, you might know these as linearization estimators or empirical standard errors. So lots of different names, all the same sort of central concept. one of the main implementations of this technique for meta-analysis is the Robometa package by my colleague Beth Tipton, Zach Fisher, and Zipeng Hu. This package came out shortly after the paper that introduced RVE to the meta-analysis community, and it's gotten to be pretty popular since then. It has this kind of big constraint in it, which is it has two working models, both of which are very constrained. So let me tell you about those and then show you how you can sort of break free of that constraint. The two working models available in Robometa are called the correlated effects model and the hierarchical effects model. Correlated effects diagrammatically looks like this. It includes a single random effect for each study. It allows the correlation between the effect size estimates from a given study. It allows for non-zero correlation there, but then it adds in a between study random effect. So it's essentially saying there can be between study heterogeneity in the true effects, but there's not any further heterogeneity within a given study that's beyond what's explained by sampling variance. The other working model in Robometa is called hierarchical effects. It's what it sounds like. It treats each effect size estimate as independent, conditional on its true effect. And it allows for correlation, or it allows for random effects within each study. Here I call those V's. And then correlation or heterogeneity between studies. So there's two, we've got a structure where we've got effect size estimates that have sampling error, and they also have true heterogeneity within each study, and then there's also between study heterogeneity. The limitation of the hierarchical effects model is that it doesn't allow the effect size estimates from a given study to be correlated with each other. It treats them as independent in terms of their sampling errors. So both of these are kind of, they're neither is ideal, they're both constrained. And the guidance available in the literature was that you should just choose between these two models, pick whichever one sort of fits the structure of your data best, even though neither of them will really fit perfectly. So my main message today is that you don't actually have to choose between these two working models, neither of which is really going to capture both of the all of the main features that of the types of data that you encounter in practice. A different strategy is to combine models estimated with metafor with the clubSandwich package for robust variance estimation. So in particular, metafor includes a function rma.mv for multivariate that provides a very versatile set of random effects structures. You can fit multi-level random effects models, you can fit multivariate random effects models, you can fit multi-level multivariate random effects models, and all sorts of other acronyms to your heart's content. Using the workflow that I'll demonstrate, you can treat the models estimated in metafor as if they're working models, and then do inference based on RVE methods. One particular sort of leading candidate for a working model is what we describe in the paper that I mentioned at the top as the correlated and hierarchical effects model, essentially mashing up the previously available correlated effects model and hierarchical effects model, just combining all of the features into one working model. So this model lets you allows for correlated effect size estimates. If you're not able to get accurate information about the degree of correlation, you can still use a rough guess about the degree of correlation. As long as you're in the ballpark, this sort of working model will still sort of be beneficial. And the other feature of this model is that it allows both for within study heterogeneity and between study heterogeneity. So you don't have to ignore one of the levels of your data structure. So this gives us a structure that looks like this, where we've got correlated effect size estimates. So we're allowing for dependence between in the sampling errors for estimates that are drawn from the same study. We're also allowing for heterogeneity within study with the V random effects, and heterogeneity between study with the U random effects. And if you want so the estimating equation here looks like this. We've got effect size estimate I in study J modeled as an overall average effect, a between study random effect UJ, a within study random effect VIJ, and then sampling error EIJ. And we allow for correlation between the sampling errors within a given study, assuming that there's just some correlation that you specify, some value rho describing the correlation between effect size estimate EHI and EIJ. I'm sorry, correlation between effect size estimate EHJ and EIJ. So two estimates that are nested within the same study. So if you fit this model in metafor, the standard errors and confidence intervals, that hypothesis tests that come out of metafor will not be robust. They will only really be valid if your working model is exactly right. So if some aspect of this model is misspecified, then you may not have the same degree of confidence that the test statistics are really valid. To remedy that, we can combine metafor with clubSandwich. So clubSandwich is, it's actually a fairly general package that provides robust standard errors, hypothesis tests, confidence intervals for a number of different types of models. So linear models, multi-level models, and it has sort of seamless support for models generated by metafor. The particular advantage of using the clubSandwich packages is that it includes some small sample corrections that give you an extra level of insurance when using robust variance estimation. So regular RVE methods are justified on the basis of sort of asymptotic approximations. So to work well, you have to have a sufficiently large number of clusters, or in our context, a sufficiently large number of studies in your meta-analysis to be able to trust regular robust variance estimation methods. And actually, you can get regular robust variance estimation through the metafor package. There's a function just called robust that will spit out robust standard errors. But the function in metafor doesn't include the same type of small sample correction. clubSandwich has this, so it uses small sample sort of adjustments that will make the standard errors closer to unbiased, even when you have a small number of studies. And it also uses some adjustments to the hypothesis tests and confidence intervals to give you, based on essentially using T degrees of freedom instead of using large sample approximations. So all of this gives you sort of extra insurance that, that the methods will work well, even with small samples, and even if your model is misspecified to some extent. Here's just a quick demonstration of the workflow. So if you load up metafor, load up clubSandwich, and then I'm going to use the data from the Tanner Smith and Lipsey meta-analysis. The first step in this workflow is to create the sampling variance covariance matrix using some assumption about the degree of correlation between effect size estimates. So the impute covariance matrix function from clubSandwich will do exactly that. The syntax here, I'm saying I'm taking the sampling variances, specifying a cluster ID, and then specifying an assumption about the degree of correlation of effect size estimates nested within a given study. So here I'm making this kind of simple, rough assumption that the effect size estimates are correlated at 0.6. Once you've got this variance covariance matrix, you can feed it into metafor using the v argument in the rma.mv function, fit whatever model you're interested in fitting. And if you store the result, here I'm storing it in mod. And then, I'm sorry, I'm having trouble with my interface here. Once you stored the result in an object, you can feed that fitted model into the clubSandwich functions to get robust variance estimates. So there's clubSandwich has a function confint that will give you cluster robust confidence intervals. So I would just say here confint of mod, and I specify the variance covariance matrix as CR2, the CR2 variant gives us these small sample adjustments. And out will come robust confidence intervals. So to sum up, why use this particular combination of packages? Why use this particular workflow? There are a couple of advantages. One is that using these more flexible working models will let you better approximate the real dependence structure of your data. That's advantageous because if you get closer to the truth, you'll get more precise estimates of average effects or more precise estimates of your meta-regression coefficients. So as in smaller standard errors, folks, like put in some extra work to better approximate the real dependence structure, and you get gains in terms of more precise estimates and inferences. A further advantage is that using these more flexible working models gives you a better description of the structure of heterogeneity in your effect sizes. For example, you can talk about both between and within study variance in the true effect sizes. That's useful just descriptively and as a guide to model fitting. If you know that if you identify, for example, that you've got a lot of within study variation in the effect sizes, you can look to, say, find some additional moderators that explain that within study variation to cut down, right? Yield a better predictive model. And then using clubSandwich provides a degree of insurance or protection against model misspecification. And it also provides protection against these sort of small sample concerns. I'll mention too, so the methods in clubSandwich are quite general. They can be used with a variety of other multi-level models and linear regression models. So clubSandwich includes methods and will work pretty much out of the box. If you're fitting just a regular linear regression model, a multivariate linear regression model, generalized linear model, instrumental variables analysis with two-stage least squares. It also works with Elmer from the LME4 package, or with LME from the NLME package, both of which will give you let you fit quite complex hierarchical linear models or linear mixed effects models. If you want more of the technical details, if you're looking for examples of how to apply this in practice, code, if you want to dig into our simulation evidence, you can find all of that in this paper. There's also a preprint available on my website. All of the supplementary materials and replication materials are also available there. So I'll leave it there, and I'm happy to take questions.
All right, thank you for this really nice and clear presentation. So if there are any questions, you can post them to the chat, or you can unmute yourself if you are comfortable with that and ask your question directly. I haven't seen any questions in the chat so far. That's probably because the talk was so crystal clear.
Hello, I have one quick question. So when running data through the clubSandwich after the metafor, how can we check between and within variance? Can we interpret that with the same like a Q-test or from the metafor, or does clubSandwich give separate metrics for the within and between variance?
That's a really good question. And so to examine the heterogeneity estimates, like between study variance and within study variance, you'll have to inspect the fitted metafor model directly. clubSandwich doesn't give you separate estimates of those variance components. It actually just uses those variance components. It takes them as given. And because it's treating them as a working model, it doesn't do any sort of inference about the variance components. This RVE methods, at least as they're currently developed, are really only about doing inference about average effect size estimates or about the coefficients in a meta regression.
Thank you.
I see another question in the chat. Is it possible to estimate prediction intervals based on RVE. This is another, this question has been coming up a lot on the mailing lists, and I've gotten it a couple of times. I don't have a great answer for it because the prediction intervals involve both the estimates of heterogeneity, like the between study variance, and they involve the estimates of the uncertainty about the average effect size. So because you need to use both of those to construct a prediction interval, it seems like you need to get the model right. You essentially have to have a correctly specified model to generate a valid prediction interval, at least based on the methods that I'm aware of. So mechanically, you could use robust variance estimation methods to estimate, to get like a standard error on the average effect size estimate. So that part would be robust, but then the heterogeneity estimate is not necessarily robust. So the logic of sort of combining a robust method and a non-robust method to create one prediction interval is not entirely clear. I don't know of any methodological work that's looked at this. It's definitely sort of a direction that would be useful to investigate further. Wolfgang, do you have any? If you have any thoughts on that, I'm sure you've pondered it also.
It's a very good question, and I think, yeah, just what you said. I think at the moment you would have to strive to get your working model to be a good working model, so you get somewhat trustworthy estimates of heterogeneity at the various levels, or whatever your working model is. And then combining this with robust variance estimation, at least you have a pretty good estimate of the standard error of the average effect, but you need a good working model to get these prediction intervals, at least given the methodology that's available right now, I would say. So,
yeah, I would say, just add to that, I think there hasn't been much methodological work looking at the performance of prediction intervals for any of these so more complex multivariate or multi-level meta-analysis models. If there's any doc students in the audience looking for research topics, have at it. It would be really a useful contribution.
If I may tag on a question here, actually this sort of came up on the mailing list just today, and since I have you on the line here, I can just ask you directly. So the issue was the following: if you have a working model that you think is maybe some decent approximation to what's going on underneath, and then you use robust variance estimation, and you get sort of a new standard error and a new test for the average effect. And so now you compare the sort of unadjusted test, or the test that you get from your working model, with the results that you get from robust variance estimation. Do you think that the degree of discrepancy between those two sets of results tells you something about how bad your working model is? Can you use this to sort of gauge whether your working model is a decent approximation or not? I'm very curious about your thoughts on this.
Yeah. It's an interesting question. I think yes. In fact, there is work in econometrics in practically a very different context, but technically or formally a very similar context that develops tests for model misspecification along these lines by comparing like a robust or cluster robust variance estimate to a model-based one. But I think one of the takeaways from, sort of as an informal guide to whether you've really like screwed up your working model, I think that's a reasonable thing to do. One of the takeaways from that literature, though, is that the tests like that are sort of not that useful formally because they have limited power to detect misspecification. So if you're using a test like that to decide whether or not you can trust your model-based inferences, using it as a formal pretest is going to like screw everything up anyways. So it's not really a good idea as a sort of formal model misspecification step, or specification check or pre-testing step. If you've got, if you have epistemic doubts about whether you've got the correctly specified model, you should probably just buy the RVE insurance in the first place.
Great, thanks. And there's another question in the chat if you want to take a look at this.
Oh, so the question is, do you have any recommendations about how to guess the degree of correlation in the effect size estimates? Another really good question. So in some cases, I would say that sort of like the best thing to do, the gold standard approach would be collect what information you can about the correlation between effect size estimates. It may be that some of the studies give you the information that you need, and you can use that to inform your guess about the correlation, even in the studies where the information is not available. Short of that, maybe you use your context knowledge or your subject matter knowledge about the outcomes that are involved in your synthesis to make a sort of ballpark guess about, say, for example, how correlated internalizing symptoms and externalizing symptoms are if your effect sizes are all about depression. And then it's also very common and recommended to pair these methods with some sensitivity analysis, looking at a range of different correlations just to check whether your bottom line conclusions are sensitive to the assumption that you make here. Typically, we find that the methods are quite robust to this assumption. The estimates and robust inferences don't really vary much depending on based on what correlation you use. Although the variance component estimates between study heterogeneity, within study heterogeneity, will be somewhat more sensitive to your assumed correlation. So it's good to do sensitivity analysis on this sort of thing.
All right, thank you. So we're going to move on